Welcome back to the OpenHPI course Code of Life. Today we want to solve the puzzle. You have learned so much in the last days and weeks, and now we want to bring all these individual puzzle pieces a little bit more together. First of all, I'd like to give you an understanding that DNA data and genetic data is only a small portion of the whole world of medical data. We are talking here about two challenges in IT. This data is typically distributed, stored at individual sites, and individual partners or departments are creating them. And the second thing is they are highly heterogeneously. The challenge here is really they have different formats, and you see here some examples for them. Like PubMed is one database for publications. This is literature, this is text. You see the human genome data, we have already talked about this in the course. You have hospital information data, which is structured, of course, in a relational format. And you have informations like prescription data, maybe clinical trials, which is a huge text, but also more and more sensors on intensive care units, but also wearables which send around short data, but at a high frequency. The challenge here is, again, the doctor in the middle, how to bring all this information together. So therefore, we want to talk at least a little bit about the requirements, software requirements in life sciences. The most important requirement is today the time. Time matters in the clinical context because the earlier you come to the right data and identify the right portion of data, the earlier you can start or adapt an existing therapy. Therefore, the real-time data analysis is very important. And the second important thing in life sciences, everyone is looking for so-called maintained software. That means you do not want to have a kind of CD or any kind of version you have to install, configure by your own, and every time you have an update, you have to start from scratch. The IT department is looking for something like a maintainer who is solving all these issues remotely. They want to get the newest technology to their doctors and researchers, but they don't want to take care about all the configuration and maintenance stuff. So in life sciences, especially in the clinical world, you have a lot of restrictions which are important to understand. We are talking here about data privacy. Data privacy, this is sensitive information. It should never leave the hospital site or leave a certain kind of campus. Therefore, you have to ensure this by certain measures. Therefore, data locality is very important. Today, no one is really interested in uploading any kind of sensitive information from a clinic into a public cloud service. And last but not least, we have talked about this, the volume of the data is a challenge. At least for the genomic data and imaging data and proteomic data, they are huge, they are really huge. And even with the fastest internet connection, it takes hours to transport them from A to B. So what could be the solution here? And we propose something which is called the federated in-memory database system. And we want to compare this a little bit with the existing approach of cloud computing. Therefore, let's have a glimpse of how current alternatives for cloud computing look like. The first thing is I brought you here in a pretty easy image. This is a model in fundamental modeling concepts. And what you see here, you have on the left-hand side, side A, let's say this is a hospital. On the side B, there's a department for pathology or a wet lab. And these both sides have a local system, a storage where the data is located, and you have some kind of synchronization services. And in the middle, you see cloud service provider A and B. So that means the storage, the local information and data will be synced to one storage provider or cloud service provider, and then they sync it again in between. Of course, the network connection between this cloud service provider might be much faster and better than between the sites. However, you see you have to replicate the data, so to say. And the first time you will create data in your local system, then a second time in the service provider A, and then in B, and then it comes to site B. This is time consuming. This is really a challenge because you multiply also the volume of the data by doing the replication. And important to say, today sharing typically involves copying. So sharing data is typically resulting in copying data, and therefore you have some challenges like how to handle concurrent versioning changes, synchronization in this term, but also the matter of fact that you lose control about your data. Once you have copied it, everyone can do whatever with it without your further notice. A second approach, and therefore it's really a glimpse idea of how it looks like. A second approach could be both sites externalize, outsource all their local IT systems to one and the same cloud service provider. So typically the people on site A and B now no longer access any local system, but they directly access the cloud system. The cloud system uses the same cloud storage and have maybe one cloud synchronization service with other partners. So in the end, this is at least reducing the number of copies in between and reduces any kind of information loss. However, you have to keep all the data, all the sensitive data at this cloud service provider. And the challenge here is single point of failure. What happens if you cannot access the system, and so on and so on. So our federated in-memory database system is going a similar hybrid way in between these two worlds. So we try to combine here the private and the public cloud by enriching this kind as a managed service provider. So in the middle you see the cloud service provider, and the cloud service provider is really keeping only the applications, the algorithms, but also the publicly available annotation data and all that stuff, and provides them to the local sites. The local site, again, has a local in-memory database system, and this is where the system also works. So all sensitive information is kept locally, is stored locally, and only the algorithms and the code is sent to each of these sites. That means, in contrast to the other versions, you have still full control. You never lose the control about your primary data, and sharing does not mean copying. For instance, if site A wants to give access for certain kind of data to site B, what happens? They will share the pointer to the data, and through that infrastructure they can access this. And this infrastructure is not a publicly on the internet available infrastructure. This is built around virtual private networks. That means these sites are associated and it's not publicly exchanged over the internet. So this is the fundamental idea behind the federated in-memory database system. And how does it look like in our context of the Analyzed Genomes framework? First of all, we took this in-memory database system, and now I'd like to abstract it a little bit. So we are talking about one federated system, and I don't want to point out the individual sites any longer. What we do, we bring in all the relevant data, the data which is important for the clinicians, for the oncologists, and so on. And therefore, we talk to them, we go to them in interdisciplinary teams. We want to understand how their processes look like today. And once we have this information, we ask them, is it possible that this and this task might be done automatically? And they typically say, yes, of course, I have to do this because there's no better alternative at that point in time. So therefore, we acquire this data, and this is typically the Google approach, right? We index the existing data, we monitor the sites, because typically there are versions released on a regular basis. For instance, genetic annotations are released every few months, and then you keep them here at a single point up to date. In this fact, it's kind of copying the data, because many of these primary sites have one challenge. They are not really 100% available because they are research sites. They have throughput issues. If a certain daytime, a lot of people work with them. Here we have a solid copy, and if you access them, you have even the chance to work with the data if the primary source is currently not available. And on top, and this is important, still within the in-memory database system, we created these very specific extension for life sciences. Applications, tools, statistical methods, and all that stuff, which is now part of the in-memory database system and can be consumed by our applications built on top. And I don't want to talk about the applications too much, but you see on top this red bubbles, and these red bubbles indicate our API, our interfaces, where existing applications use the tools. It's typically a JSON interface, and based on this JSON format, you can also attach them to your existing processes. You do not have to use these existing applications. You can also implement it in your existing process. And now I'd like to introduce to you some words about the in-memory database technology. Typically, I refer to this as a toolbox. If you call, for instance, an electrician because your power went down, they have a toolbox with them, and they have certain tools to identify where's the problem. And this is also a toolbox, and it's a bit misleading, the term, because it's more than a database. It's not only putting data into a system and being able to query the data, it's really providing you certain tools. You can run statistical analysis on top of it, you can distribute the data, but also you can do a lot of more important things which are typically out of the scope of a database. For instance, you can do text mining with that. And based on this tool set, which is our technology foundation, we build very specific applications together with the experts from the individual disciplines, like medical doctors, oncologists, but also researchers. So now I'd like to introduce some of these applications to you, because you will also have the chance to use them in a hands-on experience exercise this week. First of all, I'd like to talk about the Genome Browser. The Genome Browser is one tool which combines the the processing of the genetic data, which is in a digital format, from the FASTQ file to the variant, the list of variants, and the annotation process. This tool is very important because it is a one-shop solution. You can start with your FASTQ file, prepare your standardized processing pipeline, press the play button, and you get in a very short time, you get the results annotated with the global available knowledge. And therefore, this tool was also honored 2 years ago, 2014, with the European Life Science Award. Although they typically honor inventions in the field of medical products or medical devices, they are really impressed by the usability and the improved process here. The second example I brought here is the so-called drug response analyzer. This project is very specific for the oncology field, and you see some partners here. It builds on 3 big data sources. First of all, patient data, which is typically stored in the hospital information system. It's data about the smoking stage or the age or any kind of medication in the back. Then you have tumor-specific data, and we're talking here about genetic data, which characterize one of the tumor. And on the right-hand side, the compound interaction data. That means these tumor samples, and this is important here to know, these tumor samples were taken and in so-called xenograft experiments, they were sent into mice, and the mice was treated with a certain kind of medication to see the response of this specific tumor on a certain medication. Although these processes, this kind of lab work with the xenograft experiment is time-consuming, that's the reason why we invented this kind of library. This library gives you 2 possibilities. First of all, you find a 100% match of the tumor and the patient specifics in the library, and then you can easily refer to historic case. Here we have the proved evidence that this and this kind of therapy works good or works bad. Typically, you do not find this 100% match. Therefore, this tool enables the interpolation of the data, which includes all the very similar patients and gives you a kind of assessment of the expected therapy success. This is important for clinicians because now, of course, they can take any kind of chemotherapy, like let's say they have 10 available at a certain time, but now they can start with one very particular because they do not have to trust only their experiences or their gut feeling. Now they can say, okay, here's a calculation which gives us a hint for this or this therapy, and here are some bases, some foundation for our decision of historic basis. This is important for clinicians. The next example, which I'd like to introduce to you, is the so-called medical knowledge cockpit. And the medical knowledge cockpit, again, is for patients and clinicians. Here you see one example. On the top right side, you see the electronic medical record of this patient, Jane Doe. You see some information which are extracted from the electronic medical record in the clinic. And on the left-hand side, you see it's very similar to Google. You have a search field and you can enter some terms. And typically you enter here terms like gene name or indication. And in this case, we have again our patient, our lung cancer patient. You could easily enter all the biomarkers to get results. And how does the information look like for clinicians? For instance, they get access to pathways. Pathways model the interaction between genes and the effect on the human body. It's important to understand if a gene is altered, what effect they could expect in the human body. Furthermore, they have access to latest clinical trials. And that means if you have a patient, you will not look for a clinical trial, but it proposes it to you like a shopping system at Amazon, for instance. Did you know patients like you are eligible for this and this clinical trial? And this is helpful because if you have no alternative, you will go for the latest medical knowledge, which is available only in clinical trials. Last but not least, you will get also access to latest international publications. And you see here, it's a PDF preview of one of the publications, and it automatically highlights the relevant terms. So that means you do not have to screen all the content within the PDF and read it from the first page to the last page. It is doing exactly what researchers are doing. You focus on the specific regions where most of your terms are mentioned. You can read this short paragraph and then you can decide to follow up with this publication or not. So what to take home from today? First of all, medical data is much more diverse than we have seen so far. So the genetic data is just a small subset of all of that. It's very important to enable also the linkage and the real-time analysis of this data in the context of a treatment and on the clinical environment. And therefore we need specific tools, and these specific tools need to be developed together with the subject matter experts, clinicians, oncologists, and doctors. And you will have the experience now, and you can play around with some of these in our exercises. Go to analyzegenomes.com and have a test drive on your own and have a hands-on experience how they work. Therefore, I'm really interested in your feedback and I'm looking forward to see you next time.